Well, good afternoon, everyone. I am delighted to be able to join you and introduce today's speaker for the Wednesday afternoon lecture series, albeit done virtually. This is Leonard Zon, my friend Len, who I've known for quite a few decades and who's doing remarkable work on hematopoiesis and cancer. The title of his presentation, Blood Stem Cell Clonality and the Niche, probably has attracted your curiosity. What niche is he talking about? Well, I think we're going to find out. So let me say a word about Len. He got his undergraduate degree at Muhlenberg College, which probably not too many of you would know about, but apparently got him off to a pretty good start, considering what happened subsequently. Got his MD at Thomas Jefferson in Philly, and then went to Boston, where he's kind of been ever since. First as intern and resident at the Deaconess Hospital, then as a fellow in oncology at the Dana-Farber, and then as part of that, as a research fellow in Children's Hospital with Stuart Orkin. By 1991, he became an assistant professor of pediatrics. Not long after that, an assistant investigator of HHMI, and over the course of the coming years, became associate and then full professor of pediatrics, now Grousbeck professor of pediatrics, and continues to be supported by the Howard Hughes Medical Institute. He is also a professor of stem cell and regenerative biology at Harvard Medical School and an associate member of the Broad Institute. He's been recognized by a number of awards. We're happy to say NIH is part of that. He got a Physician Scientist Award from NIH some time ago. Has been elected to the American Society for Clinical Investigation, in fact, served as president of the ASCI. He's a member of the National Academy of Medicine and the American Academy of Arts and Sciences. He received the E. Donnell Thomas Prize for the American Society of Hematology and the Donald Metcalf Award of the International Society for Experimental Hematology. And again, let's highlight NIH's role. He got the Alfred Knudsen Award from the NCI in 2016. So his work over these years has been groundbreaking in many ways, but I think perhaps many people know him perhaps most prominently for the way in which he has introduced and convinced many of us to utilize the zebrafish as a really remarkable model system for understanding an awful lot about development, stem cell biology and cancer, where he has used that both to understand vertebrate blood development, but also treatments for patients with melanoma as well as blood disorders. And yes, over these years, progressively moving this more and more into sophisticated understanding of the nature of those malignancies that arise in the hematopoietic system brings him to today, where I think he's going to tell us quite a bit about how those things get started, how blood cell clonality occurs, and what we might even start to think about doing about it in novel ways that would not have been apparent without this kind of research. So again, as I mentioned, Len and I have been friends, and it's wonderful to have him come to NIH today to give this Wednesday afternoon lecture. I appreciate all the people that have linked up to hear this. I will apologize that I won't be there at the end to handle the Q&A because of a congressional meeting that I have to have, but I'm looking forward to listening until I have to jump into that other role. And so without further ado, let me welcome Professor Leonard Zon, who's going to teach us about blood stem cell clonality and the niche. Len, it's all yours.
Great. Well, thanks very much, Francis. It's really a pleasure. I thought I would throw this picture up. I had the privilege, this isn't a medical meeting, but many of you who are probably on the call know these folks. But this was our band. And the Doc Rock Band, and we played about 9 years ago at the Celebration of Science, where we were trying to save the NIH from sequestration, if you remember, Francis. So,
oh my God, yes.
So I told Francis now that he is taking a different position that we need to take the band on the road. So hopefully that works.
I'm here.
I've started a number of biotech companies, and I just wanted to put them in as disclosures. They don't relate at all to my talk today. So I have focused my entire life on studying the blood system and particularly understanding how stem cells self-renew. So what happens here is you have a stem cell and it migrates into the marrow, into the bones. And this stem cell can self-renew or it can differentiate into the lineages of blood cells. Now, we know that this process works incredibly well because we can do a bone marrow transplant on a patient and rescue their blood system for a lifetime. And you only need one transplant for a lifetime. Now, this process is facilitated by the niche. This includes the vascular endothelial cells of the niche, the stromal cells, which are specialized fibroblasts, and even the bone cells participate in allowing these stem cells to self-renew. Now, even though we do bone marrow transplants every day, still about 25% of patients die from their transplants. And I think studying this niche and understanding how stem cells self-renew is something that would be very helpful therapeutically in the future. Now, we use the zebrafish as a model system to study this process, and I'd like to show you how blood stem cells are born. So this is a 36-hour embryo. And we're going to dive into the dorsal aorta where the stem cells are actually born. So when we dive in, what we see is an endothelial cell that is hemogenic. That is, that it's going to make a blood stem cell. It forms a hammock and rounds up, and that's a blood stem cell. This occurred during embryogenesis, and that stem cell is then going to go off into circulation and colonize the sites of hematopoiesis in the future. In the first site, in the human would be in the fetal liver, but in the fish, it's in this region in the tail called the caudal hematopoietic tissue. And this stem cell stops on an endothelial cell, transmigrates into the niche, the environment, doubles, and then some of these stem cells can then go off into circulation and colonize the marrow. Now, the marrow in the zebrafish is in its kidney. In humans, it would be inside the bones. And this makes the kidney a hematopoietic organ in the fish. Some of the cells bypass the kidney and go straight to the thymus. This starts the immune system of the fish. And all of this lets this animal have blood really throughout its entire lifetime. And certainly one of the powers of the zebrafish is the great ability to watch all those things live. Several years ago, we published a fish that had green stem cells. We used an enhancer of the RUNX1 gene that is specifically expressing in the stem cells that are born in the aorta. These then go off in circulation and then they land in the caudal hematopoietic tissue. And I've always wondered what it looked like when a stem cell landed, particularly when I do a transplant on a patient. Unfortunately, we're all opaque, so I can't see what happens in the patient. But in the fish, if you follow this circle here, you'll see that the stem cell transmigrates outside the blood vessel system. But then we saw something very strange. The blood vessels actually wrap themselves back around the stem cell in a process we call endothelial cuddling. There's a division, a turn, and one stem cell leaves and the other one stays. And this is a very stereotypic behavior. We're able to take that animal and actually send it for correlative electron microscopy, producing the highest power view of a stem cell niche in a vertebrate. And we got some surprises. Here's this stem progenitor cell. It is surrounded by 5 endothelial cells forming a pocket. But this stem cell that was traveling all around circulation lands and attaches to a stromal cell. And it's on the surface of the stromal cell that the stem cell will actually divide. So this niche became really interesting to us, and we wanted to understand it at a better level. And Elliot Hagedorn, who I should say is now an assistant professor at Boston University, did this set of experiments. He did something called Rna Tomography. What this does is to take this region, which is going to be the blood niche, and he sectioned across the niche. And then every section went into a tube for Rna-Seq. This allows a spatial evaluation, and you're able to see genes like that are epidermal genes on the outside of that tail, top and bottom. And for us, what was very exciting is there were genes that were only expressed by the niche and not even by the stem cells, just by the niche. So this made us wonder what's so special about this particular set of genes and tissues in the niche. So it turned out that 29 of the genes were expressed in the blood vessels. And we knew this because when we had done our own Rna-Seq experiments, a number of genes had shown up in this KDRL, which is the KDR receptor, the FLIC1 receptor. And so we could sort these cells and show that these genes were there. But when we started to do in situ hybridizations to see where the RNA was expressed, we got some really interesting information. So here's, again, this KDRL gene. This is a gene that's expressed by all endothelial cells. And you can see the intersemitic sprouts, and all blood vessels are expressing this. But these 4 genes here are just examples of the 29 that we saw that were in the blood vessels. These are only expressed. You can look at this fish here, you know, there's really no expression except in this niche where the blood cells are. Same for this one, GPR182. So these turn out to be the specialized venous sinusoids of the marrow. And we're able to find a molecular signature for these based on these analyses. So I wondered, how do you get this kind of expression pattern? So what we did is we took the promoter of 2 of the genes, E-selectin or a mannose receptor, and we drove green fluorescent protein in the background of a pan-endothelial red marker. And you can see how beautiful this is. In this particular transgenic of 1.3 kilobases, only this region lights up. So to me, that indicates that there's a specific code of some form that is telling these particular venous cells to acquire its fate. And we decided to go after the code. So first, what we did is HHMI on sorted populations of cells. And what you can see here is the niche-specific endothelial cells have an open chromatin on this gene stabilin 2, but nowhere else on any of the other endothelial cells. We took this little peak here, which is 400 base pairs, and coupled it to green fluorescent protein and injected fish. And the only place we saw the green was in this niche endothelial cells. So this is a very specific effect. And we started studying other peaks and got pretty much the same result. If we take these peaks, they drive in this caudal hematopoietic tissue. So basically, 12 out of 15 cloned peaks drove expression in these venous sinusoids. Now, what if you look at peaks that are pan-endothelial? So they're expressed in all the endothelial cells. Well, those drove in all the endothelial cells. So again, suggesting that there's a code of some form that's responsible for making these specialized venous cells. Well, what's the code? So with HHMI, you can look at motif enrichment. And we found that there's an ET site, a SOC site, and a nuclear hormone site that's particularly open in this, or used in this particular endothelial population. As opposed to when we look at the pan-endothelial population, it's really just the ET sites that are really involved. So we decided to look at what transcription factors were specifically expressed in these niche endothelial cells and had the opportunity to bind to those motifs. And what we could see is that there were 7 transcription factors, 3 ETS-related transcription factors, 2 SOX family members, and 2 nuclear hormone receptor family members. So what we decided to do was to see if those factors had the ability to reprogram venous sinusoids in vivo. So we took a ubiquitous promoter and drove all 7 genes by overexpression. And we looked at one of the markers, either MRC1 or E-selectin. And as you can see, compared to controls, if you look at the 7-factor injection, we start getting extra veins, essentially. And if you look here at one of our transgenics with our MRC1 GFP, the vein-specific GFP reporter, you can see even an extra vessel that's normally not there, but it is a venous sinusoid. So then we did the kind of Yamanaka trick where we subtracted singles away from the 7, and we're able to find out that if you express members of the ETS family, ETS2, ETS-SOC7, or NR4A1, that you could get a pretty high rate of these extra vessels. And so this was our most efficient combination, and so we studied that most of all. So Elliot, came to me one day and he said, I've done this experiment and I think this is the best day I've ever had as a postdoc. And I said, well, what was it? And he said, well, I did this in the background of red stem cells. And this is a little ectopic niche that was formed. And I hope you can see that the red stem cells left their normal niche and went to the new niche. So I thought that was super exciting. And I said, well, do they cuddle? Because I feel cuddling was one of the functions that's quite important. And he says, yes, I see cuddling. And I said, well, do they divide in these new niches? And he says, yep, they divide. So this is an animal, and this is the normal niche, and you can see a number of stem cells here. And in this particular animal, we've been lucky enough to capture a little ectopic niche. And you can see it's attracted these stem cells there. And if you focus on the top here, what you'll see is those stem cells go on to divide and are even released. So this is a fully functional niche that's been reprogrammed. And I think that's very exciting. So we wanted to see, this is a fetal niche or a larval niche. We wanted to see if this was true in the adulthood. We ended up doing single cell analysis on the adult niche, and many of our genes were present there. And then we collaborated with Shaheen Rafi's lab, who had taken endothelial cells from many different organs through development. And what was interesting is our signature of genes was very high in the fetal liver in a mouse. It kind of went down in the liver as the animals were born, and many of you know that the liver is not a hematopoietic organ in the mouse later in development. And where we see the signature come back is in the bone marrow of the adult. So it seems that the signature of genes, these venous sinusoids, are present in the sites that are actually hematopoietic. So in this particular story, we were able to find that Rna Tomography allowed us to find a set of genes that were venous sinusoidal specific. And then we're able to find a code, an ETS, SOX, and nuclear hormone receptor code, and then be able to reprogram these cells to in vivo in a niche. And the use of this could be very interesting for patients who have myelodysplasia, because their stem cells, once the marrow fibroses, they don't know where to go and they don't do very well. And so if we could actually take iPS cells and reprogram them into these venous sinusoids, maybe we could inject them into the arm of a patient with MDS and make those patients then have a new niche where the stem cells could go and it would rescue the patient. So we've been playing with iPS-derived veins and seeing if we can reprogram them. And we have some interesting preliminary data, but hopefully at the next time I give a Walls lecture, I'll be back to tell you about those studies. So now what I'd like to do is to talk about the stem cells and think about clonality. So what happens here is most of us have polyclonal blood. And that means that when you had the stem cells born in your aorta, when you were an embryo, you generated a certain number of clones of stem cells, and that is reflected in your adulthood. Now, as people age, a number of investigators have found that there are mutations in the peripheral blood of aged individuals, and these mutations often are in epigenetic regulator genes. And this is a theme that's going on for many different tissues. If you think about eyelids, the wrinkles in your eyelids have mutant P53 clones. If you think about the esophagus of old folks, the esophagus has clones of Notch mutations. In the intestine, there are clones of cells that are precancerous, but they exist. And so in these patients who have what's called clonal hematopoiesis of indeterminate potential, they acquire mutations in one of their stem cells. And then the blood of this system, of these patients, about 20 to 30% of the blood comes from a single stem cell. And this predisposes you to myelodysplasia or ultimately to leukemia. So we would like to understand this a little bit better using our zebrafish model. To do this, we needed a method to clonally track the stem cells. And we came up with the idea, and this is John Henninger and Serena Vagian's idea, to actually color each stem cell a different color. So what we do is we have these zebra bow or rainbow type of fish. These have red, blue, and green plasmids, about 15 to 20 plasmids all over their genome, and they're separated by LOX P sites. If you now come in with a blood-specific Cre-ERT2, you will start rearranging these plasmids and essentially dial up any color of the spectrum. We do this during embryogenesis in an inducible fashion by giving tamoxifen. And then we can grow the animals up to adulthood. All of the embryonic blood that's colored is gone by then, and only the blood that comes from stem cells will be available. And so the number of colors that you see in the peripheral blood will be the number of stem cells that were induced during embryogenesis. And so this is an embryo, and you can see it's got multicolored blood. And if you focus on this particular cell here, that's in an extravascular location. So it's probably a blue-green stem cell. And when that blue-green stem cell grows up to adulthood, it will make blue-green blood. So we grow this animal up for 3 to 4 months. It's an adult. And then we do a peripheral blood smear. And that is the peripheral blood smear. Now, as Francis can tell you, I'm a hematologist, and I think that's the prettiest blood smear I've ever seen. So if you look, the number of colors is, if you do it at 24 hours of development in a fish labeling, that's just before the birth of blood stem cells, there's 21 colors here. And so that would mean there's 21 clones of stem cells that start the blood program. You never have more stem cells being born. They're always just amplifying from those populations. And that's just shown here at 24 hours, there's 21 clones of stem cells. So now we can start to think about modeling those clonal hematopoiesis disorders. And said in one way, if you go to the human, you normally have polyclonal hematopoiesis, but you can acquire mutation that ultimately leads to adult clonal expansion. This occurs during aging. In the zebrafish, we can color every stem cell a different color. We can target genes or overexpress genes. And then what we can look for is clonal dominance in our adult fish, thereby modeling the disease. So how do you go about doing this? Well, I should say, first of all, what is the mechanism so far for clonal hematopoiesis? So what was known before our work was that you have polyclonal hematopoiesis, you get a mutation, and what happens is the mutant stem cell is pretty normal, even though it's carrying a mutation in an epigenetic regulator. And these cells will actually go on to make mutant neutrophils and monocytes and macrophages. But what's interesting is these cells that are mutant, the differentiated cells, are pumping out inflammatory cytokines like IL-1 and TNF. And this leads to the marrow being very, very inflamed. Then there's clonal dominance from an unknown mechanism. And then ultimately second mutations can happen, so you end up progressing to leukemia or MDS. So Serene Avagian, who's actually on the job market and I think is fantastic, a pediatric hem-onc fellow in my lab, invented this technique we call twister. And I hope many of you have played the twister game, and obviously the play on colors is there. This is tissue editing with inducible stem cell tagging via recombination. So what Serene did initially was very interesting. She injected 20 guide RNAs for most of the mutations that cause human clonal hematopoiesis. She also injected this with Cas9 mRNA and not protein. This was very important because we were able to measure variant allele fractions later. And by doing this mRNA, it's not as efficient as the protein, and it allows 50% of the stem cells to be wild type and 50% to be mutant. So you can now compete in vivo two stem cell populations against each other without a transplant. And this is a very big advance for the field. We then can do the color trick, so we can mark the clones, and then later grow the fish up to adulthood, bleed them to be able to see what clonality is over time, and ultimately sacrifice the fish to be able to get their marrow. And what's nice about this is because the dominant clone will be in a certain color, we can fact sort that color. And this allows us to look at what pathways are found in dominant clones versus non-dominant clones, which I think can have an impact therapeutically. This is a list of the genes that Serene targeted. And again, most of these are characteristic genes. You may have heard of TET2, ASXL1, and DNMT3A. These are the ones that most commonly cause CHIP, but a few others. And there were some control genes and also some pseudogenes that were put in just to make sure that, that everything was behaving. So this worked incredibly well. Over here is a control fish. You can see about 20 colors of cells, and there's really nothing dominant here. But look at this fish. It's got a massively dominant clone, and 60% of the blood is coming literally from one stem cell. So when we look at this, if you look at cluster size in this pool injected, you can clearly see that there's a lot of fish with clonal dominance. And there's another way of looking at this, Gini coefficient, which is an economics term to look at how diverse the populations are. And you can see an increased Gini coefficient with the mutants. So clearly we've been able to model CHIP. Now in our system, the gene that popped out the most was this particular gene, ASXL1. ASXL1 is an epigenetic regulator, and it's very frequently mutated in clonal hematopoiesis. Now, interestingly, when we knock out ASXL1, we do not get clonal dominance. But when we knock out exon 12, we do get clonal dominance. And it's only exon 12 mutations in humans that give clonal dominance. So there's something about this being a neomorphic allele that's specific to exon 12. But clearly we get other mutations, and it's very refreshing to see that, you know, many mutations can happen in the fish and can read out. So now we go ahead and sort the dominant clones. And here's a fish with 48% coming from one color. And again, ASXL1 really is very strong in the dominant clones compared to the non-dominant clusters. Certainly P53 and DNMT8, which is a DNMT3A ortholog, read out. But this one in our system is extremely strong. So we went ahead and then did single cell Rna-Seq analysis to see what would actually happen. And this is what you get from a marrow of a zebrafish. And it looks very, very similar to what you would see in a single cell Rna-Seq of human marrow. All the different populations are there. And we're able, again, to look at the mutant pool versus the control pool and see, you know, are there any things that are actually really interesting in terms of gene expression? So the first thing that came out, which really was blazing, is that the neutrophils and the macrophages from the dominant clone are expressing IL-1 and TNF-alpha. So that was reassuring, and that's modeling the human disease. So these marrows are definitely inflamed. But when we looked at the stem cell pools and the multipotent progenitors, we were able to see actually that modulators of inflammation, kind of anti-inflammatory factors, were present in those stem cells. This includes SOX3, a modifier of cytokine signaling, NR4A1, a nuclear hormone receptor, or another transcription factor, ATF3. So this made us think that there's a mechanism here. Perhaps these stem cells are resistant to the inflammation, and that's how they grow and double over years. And that's why it occurs in aged individuals. Said another way, here's these stem cells. You acquire mutation. This causes mutant progeny to be there. The mutant progeny make inflammatory cytokines. This is going to feed back and suppress the normal stem cells. But those mutant stem cells have anti-inflammatory factors, and that's going to make them resistant. So this also offers the opportunity that if you could disrupt these anti-inflammatory factors, this could actually treat the disease. Certainly, it's also possible to block the inflammatory cytokines, but I should point out there's a lot of inflammatory cytokines being made. So you need a couple therapies here. Perhaps getting at the seed of the bad stem cells might be useful. So Serene did this experiment, which I think was very, very exciting. So we targeted ASXL1 just by itself. And you would expect with inflammatory cytokines being there that this would lead to clonal dominance. But we also targeted ASXL1 in the presence of NR4A1 guide RNA. So in these animals, because it's a mosaic mutagenesis protocol, some of the clones that would not get NR4A1 targeting, they would continue to expand. But it would allow us to test our model to see if inhibiting NR4A1 would block stem cell expansion of the ASXL1 mutant clones. And in fish, you get a lot of fish, and you can do a lot of statistics, and it works really well. So just to give you the punchline, you can see a dominant clone in these fish, but here's one of the clones that had ASXL1 and NR4A1 knocked out, and you can see it's a small clone, and that is the result. So first of all, when we look at ASXL1 mutations, let's say deleterious indels, they lead to clonal dominance, the cluster size you can see. And then when you are a homozygous mutant, NR4A1, but an ASXL1 mutation, you see a reduction in clonality. So this would suggest, again, that inhibiting NR4A1 would block the clonal dominance. Now, if you look at the wild type situation, in other words, in some of these cells are wild type stem cells in the non-dominant clones, they don't have indels in ASXL1. The NR4A1 guide RNA has no effect on clone and cluster size. So this would indicate that there's no effect on the normal hematopoiesis, but there is a massive effect on the ASXL1 driven hematopoiesis. So it would be kind of the perfect thing for a therapy. So this twister system allows the study of blood cell clonality in the native niche. And this mosaic mutagenesis allows you to compete a wild type and a mutant stem cell in vivo. And I think there's some lessons to be learned there that transplant's a little bit artificial for measuring these things, although certainly there's a tremendous body of work for transplant. We see this inflammatory signature that's occurring in the mutant myeloid cells. And we think then that the mutant stem cells have upregulated this anti-inflammatory response, creating a situation for clonal fitness and clonal dominance. But if you're able to have a biallelic NR4A1 loss, then you would suppress this clonal dominance. There are some tool compound drugs for NR4A1, and we're testing those drugs. And so far, it looks like we might be able to suppress clonal dominance. And so this could end up being some type of therapy, although certainly if you're going to treat a patient with clonal hematopoiesis, you'd better be really, really safe because you don't want to cause any damage, because 30% of 80-year-olds have clonal hematopoiesis. And so you really want to make sure that it's incredibly safe. Maybe treating patients who are about to transform would be the population that you would want to treat. So now what I'd like to do is to tell you another news story on the influence of macrophages on clonality. And this is the work of Sam Watrous in the lab, a graduate student. Sorry. So in this particular story, we were imaging macrophages in stem cells, and we saw some really bizarre behaviors. So here is a macrophage, and you can see it intimately interacting with a stem cell. It's also removing some cytoplasmic material. And this was kind of a surprise. I'll play that movie again. What is this macrophage doing? And we thought that it was pretty frequent that macrophages were interacting with the stem cell in this kind of fetal liver equivalent area. So Sam decided to do some more intense analysis. And so what he did was to look, and remember, these stem cells were just born and then have traveled to this fetal liver equivalent, okay? And what he saw first was about 20% of all stem cells that had just been born are doomed. So they're completely eaten by a macrophage. So that was a surprise and made us think that there's some kind of quality assurance mechanism for stem cells. But then he saw this other behavior which is really remarkable. And here what we call this behavior is a grooming. And so what you're going to see here is a stem cell about to be groomed. The macrophage comes, takes a little chunk of the stem cell, and then that stem cell always goes on to divide. And this is almost as if there's a licensing event of you need the macrophage stem cell interaction to get the cell division. And so this was very remarkable, and again, makes you think about a number of questions. What's the frequency of these events? Well, I'm going to tell you it's pretty frequent. What determines the interaction between the macrophage and the stem cells? Is there anything special about these particular macrophages versus other macrophages? And what is it that's determining whether you're doomed or you're groomed? And then also, this would be a quality assurance mechanism for stem cells. I don't think has been described before. And the question is, what's being monitored? What's wrong with those stem cells that got completely eaten? And why does this even happen in the first place? Okay, so one of the things that we looked at very early on was, is it specific for the stem cells? And it certainly seems to be. So we can see the interactions with the RUNX positive cells. We don't see the macrophages interacting a lot with the blood vessels or red blood cells. And we even were able to assemble this in vitro. And again, we saw more interactions with the stem cells than the red blood cells. The percentage of cells that are groomed and doomed are roughly equal. So, and developmentally, the difference between day 2 and day 3 are basically the same. So again, this process is very frequent and happens continuously during this period until the colonization of the liver equivalent. So we wanted to see, if we could modulate the number of macrophages and see if there was an impact on stem cell clones. And to do this, we have a fish that we can treat with clodronate liposomes. These will kill all the macrophages, and you can see that here. We also have a genetic way of doing this with an IRF8 morpholino. And so you can decrease IRF8, and this will also reduce the number of macrophages tremendously. So what we decided to do is to use our zebra bow system and the twister system to be able to evaluate clonality. So again, we do our color trick, labeling all the stem cells different colors. We give clodronate at a variety of different times and see what happens to the number of stem cell clones when the fish becomes an adult. And here we got a pretty interesting result. In the sibling controls, you have those 20 colors that I was talking about. In the clodronate, it looks like you have half the number of colors in the adult. So this macrophage stem cell interaction is very remarkable at selecting clonality for the adult. And I would also say that this shows that clonality is actually determined during this early period of development. So amalgamating all of the results, if you knock down IRF8, you reduce the clone number. If you give clodronate to eliminate the macrophages, you reduce the clone number. We can do this at a number of time points. This time point was a little bit close to when the stem cells were born, so we did it later. We still get a reduction in the number of clones. And also we have a mutant and a morphant fish in ITGA4, and these have a migration problem of stem cells, but their clonality is normal. So it seems very specific to this macrophage interaction. So Sam was trying to figure this out, and what he said was it must be possible to fax purify the macrophages that have interacted with the stem cells because they still have a little chunk of red in them. And sure enough, he could fax these macrophages out and compare them to the bulk macrophages and see what's different about the macrophages that are interacting with the stem cells. Now, the other thing he can do besides like single cell RNA-seq, he can also do proteomics. And there's a core at Harvard that can do proteomics on very, very few cells, even 120 cells. And so Sam was able to look and see if there were factors or proteins that were particularly found in those macrophages that had eaten the stem cells. And what he found was this set of proteins, calreticulin, calreticulin 3A and calreticulin 3B. And this was very interesting. And I remember showing this list to Tony Green in England, and he said like, oh, that's it, calreticulin. And the reason he knew about it is that calreticulin is an ER protein, and it has a retention sequence, which is KDEL. But calreticulin can actually get cleaved. The KDL, I should say, can get cleaved off calreticulin. And this creates a calreticulin that can move to the surface and decorate glycoproteins on the surface. And Irv Weissman has been able to show in cancer cells that calreticulin can be an eat me signal for the macrophages. And this calreticulin can interact with LRP1, the receptor. It's a lipid receptor. And we can actually see that this interaction will cause some type of eat me signal. So first of all, if we look at the stem cell, we think it has calreticulin on it. The macrophages by single cell Rna-Seq are expressing the receptor for the calreticulin. It's also known that there's a bridge molecule between calreticulin and LRP1, and that is a complement, C1q. And our macrophages here, which I should say are close to M2-like, are C1q positive. So now the question is, can we look at the macrophage stem cell interactions by knocking down calreticulin? So we knock down CalR or CalR3A or 3B, and what you see are less stem cell macrophage interactions, particularly with 3A and 3B. We also could overexpress with our RUNX enhancer a CalR3A with a delta KDEL, so missing its ER retention signal. And again, if you look at the interactions, you can see more interactions if you've overexpressed this particular CalR mutant. And this is shown here that overexpressing any of the CalR mutants here, CalR or CalR3A or 3B as the KDL mutants, will lead to more interactions. So then we did the experiment of knocking down CalR3A or 3B and evaluating stem cell clonality in adulthood. And again, you can see here that there's an impact of reduced clonality if you knock down CalR3A or 3B. Now, one of the questions we had is there a specific time period where this interaction is actually happening? And what we saw actually is using our Fuchi fish, which will label the G2M phase over here, you can see that the macrophages are interacting with the stem cells as they're in the G2M phase. And then that stem cell will ultimately divide. We also can quantify this, and you can see again that if they've interacted with a macrophage, they're more likely to be Fuchi positive, they're more likely to divide, and the time to division after a macrophage interaction is within the first 45 minutes after the interaction. Now, we wanted to know more about this cell division, and we inactivated either CalR or IRF8 or control morpholino, injected, and then did EDU labeling. In this experiment, we hoped that we would see reduced cell proliferation as a result of inactivating these macrophage stem cell interactions. And that's what we saw. So again, compared to the control morpholino, if you inactivate IRF8 or the CalR3 or the CalR3B, you can see a reduction in the number of cycling RUNX positive cells. So this sets up a very interesting model, and most of the field, I think, had felt that clonality might be determined at the time of birth of the stem cells. In other words, there's a certain number of colored stem cells, and that's what you have for your entire lifetime. Our experiments shed some light on this, where actually there's probably a determinant of an adult clonality, but it's done between the time when they're born and when the colonization of the fetal liver would actually happen. One thing we could say is, you start off with these colors, which we're labeling at the time of stem cell birth. And normally what happens is you get, some clones that might double and some that don't, but everything is pretty symmetric. So you end up with all the colors in the adulthood. Now, in the case where you have fewer divisions, perhaps what happens is that you end up with selective amplification of certain colors. And then when they go to colonize, they have a higher chance of contributing ultimately to adulthood. And so you end up with a clonal dominance that's as a result, which means less colors in the peripheral blood for our experiments. So this is a clonal competition model that I think is very consistent with a number of transplant work by Cindy Dunbar and others. I think, I think we just seeing it in vivo without having to do the transplant. So we wanted to know really, where does the calreticulin have to be to see this effect? And in our zebrafish, we have the ability of making parabiotic zebrafish. And so what we can do is in an embryo that is carrying a stem cell specific marker, this one in cherry, we can inactivate calreticulin. And then over here, we can take a control morpholino and have the macrophages expressed. And then we can fuse these during embryogenesis. And then the macrophages and the stem cells can equilibrate. And then we can actually see what the interactions are and where is the calreticulin actually required. So I love these experiments. I mean, and Sam and Elliot did these, and this is really crazy that you can make these parabiotic animals that have joined circulation. And ever so often you get these beautiful ones which are kind of fused at the head region. And again, you can see the niches kind of segregating, which is really great. And again, with the macrophages and the stem cells in different colors. So the result is very interesting, and that is that the calreticulin is required on the stem cell. And this is highlighted here. This is the control injected runx morpholino, the control injected animal and the MPEG injected animal. And now if you've inactivated calreticulin, you get a reduction if the calreticulin is done on the runx positive cells. And this isn't true if it's done on the macrophages. So you need the calreticulin on the surface of the stem cells. So a model that we have right now with our work is that the stem cell is going to be colonizing that fetal liver equivalent. And it's going to come in, and these stem cells are getting ready to interact with macrophages. So the macrophages have two different behaviors. In one of the behaviors where the surface of the stem cell has a lot of calreticulin, that macrophage is just going to eat that entire stem cell. And that's goodbye to the stem cell. Now, another behavior is the grooming behavior. And in this particular situation, what happens is that the macrophage comes in and takes a chunk of the stem cell. And in this situation, the stem cell will go on and actually divide. And the way that it divides is in a very characteristic manner where it goes in those cuddled endothelial pockets and then attaches to a stromal cell, and the divisions are out perpendicular to the plane of the stromal cell. So this raises, some interesting questions that we're still working on. The first is that it allows a model where the macrophage is looking for stem cells that have a little bit of calreticulin on them. We think this leads to that partially eating behavior and ultimately to the division of the stem cells. In this situation where you have a lot of calreticulin, then this we think is meaning to be eaten. I should point out that calreticulin is known to be mutated in humans who have myeloproliferative syndrome. And the calreticulin, the mutant calreticulin, binds to the thrombopoietin receptor and stimulates the proliferation of the stem cells. Perhaps this interaction is also caused by some cytokine interaction with calreticulin, and that's something that we're looking for. So understanding how the mechanism of the division happens is something we're still trying to work on. The other aspect that we're working on is why is this happening, and what's in these stem cells to actually be able to cause this effect. And we think that there's different things the stem cells have gone through, should say stress or trafficking or mitoses. And maybe what's going on is that there's a sensing of damage. Could be DNA damage, reactive oxygen species, ER stress, or something like that in the stem cells that's being monitored. And stem cells that have too much of that are just taken out because they won't be effective later or could lead to problems later. Maybe a deficit in being able to respond to inflammation, for instance. And so these are the things that we're trying to understand about how do you present calreticulin on the surface and how do you actually allow this quality assurance mechanism to work. So that would be the end of my talk, and I want to thank the people who've done the work. I talked about Sam's work. I talked about Serene and John Henninger's work on the clonality, their recent science paper. And we have a great barcoding team of a number of investigators that are all very helpful. And with that, I'd like to thank all of you and happy to take questions.
Well, thanks very much, Len, for a great talk. And unfortunately, as you can see, Francis is now transformed into me. Rogers, I'm the director of NIDDK, and I too have been a longtime friend of Len's, and really pleased to have him be our Walls lecturer. Let me remind people that they can, if you want to send a question in, you can do so by clicking on the button below your videocast window that says send live feedback. Click on that, type in your name and question, and then we'll relay that on to Dr. Zon. And you can submit a question at any time. Unfortunately, that didn't get transmitted in the introductory materials, so there's still time. We have a couple of questions, and let me start off by asking you the first question, Len. This question is from Ron Germain, who's in the NIAID. I know you probably know Ron. Ron's question is, your correlative imaging showed the stem cells adherent to stromal cells and dividing on these stromal cells. Now, are these stromal cells ubiquitously present near the ectopic sinusoidal endothelial cells, or are they induced by the endothelial cells? And are they, in the end, not essential?
Yeah, so I would say that the stromal cells are probably essential. Mm-hmm. And what we see right now, which is really exciting, is there are, in the new niches that are formed, there are fibroblasts, probably stromal cells, that are expressing CXCL12, and they are in the region, and they kind of have an attractive mechanism to come into the niche. Mm-hmm. And so I think that it's a little more active, and we see the same behavior in this caudal hematopoietic tissue. So we're trying to understand that mechanism, whether integrins or some type of force is required to actually get them there. But it's a super interesting question, and we think it's active, and I bet that the stromal cells are actually required in some way. As many of you know, KIT ligand is made by those stromal cells that would help maybe facilitate some stem cell attachments, but other mechanisms may be at play.
Gotcha. One other point I think that Dr. Collins made, but just to repeat it, for those of you who are interested in getting CME credits, the CME code for today is 37921. 37921. I know this sounds like a word from our sponsor. We'll do our best to make sure that you get your CME credit here. We have another question in, and this question is from a Dr. Perez. And the question is, do you know what happens if you deplete complement, specifically C1a? Do you still find the same clonality in the stem cells, and do you still get grooming behavior on the monocytes?
Yeah, so we're doing that experiment right now, but we don't know the result. You know, one of the things about complement that's really interesting is like the work of Beth Stevens, where she's looked at microglial cells and how they are able to interact with synapses. Mm-hmm. And this kind of approach is really similar. And so you wonder if the complement will have an effect on clonality also. So it's something we're looking at, but we don't know the result yet.
Fantastic. You know, I just, as we're waiting for the next question, I know your lab has also obviously been at the forefront in determining and dissecting the mechanisms of melanoma. And I know that you've developed the first animal model of BRAF-driven cancer. Is any of the work that you presented today in any way related to that work on melanoma?
Yeah, so one of the things that we've been doing recently, and again, very exciting, is to study the cancer niche and compare it to the hematopoietic niche.
Mm-hmm.
And during my day, I was actually showing some of our videos, but we've been able to label CD8 positive T cells with a transgene and watch the T cells live go into a melanoma.
Wow.
And we can mount the adult fish with its tumor on a 2-photon scope and image for 18 hours.
Wow.
And when you do that, what you can see is some really interesting behaviors where these T cells and dendritic cells come together in regions of the tumor that are on the surface of the tumor. And we see really interesting interactions. And if we give a therapeutic, such as a GC oligo that's used in the clinic, injecting the tumors, we see massive changes in the tumor niche, in the stromal cells, in the endothelial cells, and the collagen that's present around the tumor. And so when we compare how the trafficking of cells is to this little stem cell niche in the embryo of the fish, there's a lot of similarities. The endothelial cells are very, very important. The stromal cells we've been able to label, very interesting. So, I'm really looking forward to doing this comparative analysis. And I think therapeutically, they're targeting these niche cells, the stromal cells and the endothelial cells, actually could have some really interesting opportunities in the future. So we're interested in those.
Fantastic. A couple of more questions have come in, and I'm going to be sensitive to your time because I know we're getting the money's worth of you. You have a number of other Zoom meetings lined up. This question is from Erica Bresciani, and she asked, do hematopoietic stem cells and macrophage maintain the same interaction that you nicely showed us in the adult marrow?
Yeah, so we don't know the result yet, but I think they're going to have a very, very similar interaction. The place where I think is most similar is when we do a bone marrow transplant, we put in a lot of cells, and we know that you don't engraft with all of those cells. So the question is there a recognition of certain stem cells that are prone to engraft based on their calreticulin levels? And could there be that selective quality assurance mechanism to select the best stem cells that are going to last for a lifetime? And so I think that the interactions are going to be similar. Again, probably during the colonization of niches and during a transplant. We're starting to image with light sheet microscopy the kidney, but we don't know the result yet.
All right, good. Here's a question from Michael Duran, and his question is, at what time does clonal dominance occur relative to the disease symptoms? This is actually a question of you as a hematologist. And at the time of disease symptoms, is it too late to attack those dominant clones that you showed? What is the state of less dominant clones at the time of these disease symptoms? He's hoping that this question is clear.
Yeah, no, that's super, super useful. So what happens is, as I said, it's 10% of 70-year-olds, 30% of 80-year-olds, and 50% of 90-year-olds have clonal hematopoiesis, have mutations in a stem cell that's leading to 20 to 30% of their blood being dominant. But about 2 or 3 weeks ago, a friend called, and her father had MDS. And very low-grade MDS, not terrible, doing very, very well on venetoclax. And what happened was the patient had TET2 mutations, but just developed an ASXL1 mutant clone. So clearly at this point, the person is on the way to having more symptoms. And so that's probably the place to intervene, is in this period of time. And a number of us are thinking about using cellular barcoding technology and variant allele fractions to be able to figure out these patient populations and ultimately do some clinical trials, perhaps short-term trials, to see if you can make clones of cells not be dominant. And so I think that's the area to actually to look in. And that, to be fair, where we found the NR4A1, you'd want to basically use an NR4A1 inhibitor in this particular patient that has that new ASXL1 mutant clone.
Fantastic. One final question. First of all, Dr. Lawrence Bazer says it's a very nice talk. Congratulate you. But he asks a sort of a general question. Can you really model MDS in the zebrafish? I think most of your answers have dealt with that, but you might want to just recap.
Sure. Yeah, I think we're pretty accurately modeling the MDS situation in the zebrafish, and we even have fish that have leukemia. And we've been doing some clonal barcoding in leukemia, and it's been very interesting. So I think we can model it. The only thing that I would say that's a caveat, and I always don't want to oversell, everything with the zebrafish, we mutate the stem cells at the beginning of their development. Right. And so then they become dominant over time, and it does take months for them to be dominant. But I do think that in a human, you already have an adult when the mutation actually happens. So it is a little bit different. The other thing I would say, which is pretty interesting, we've started to model pediatric myelodysplastic syndromes and pediatric predispositions to leukemia. And even in the science paper that we had recently, we're able to demonstrate that early in a fish's life, if you have a RUNX1 mutation, you're clonally dominant really when you're born. And so that starts to make you think of predisposition syndromes and already being dominant. And that's how the syndrome actually happens. So the mechanism of aging versus these congenital defects is actually identical, except the fact that you're born dominant in the RUNX1 mutation versus if you have acquired mutations later in life. So it's pretty interesting.
Excellent. Well, there are a few more questions, but I see your next handlers are here to whisk you off to your next meeting. And I know you have a tea at 4:15. Lynn, that was a great talk. It really covered a lot of territory, and this is such a beautiful model to actually visualize what's going on in vivo. So, on behalf of Dr. Collins, and I'm sure he would do that if he was here, let's give Dr. Zon our virtual congratulations on a perfect talk. And with that, thanks very much, and I'll tell the audience that we are now adjourned. But I want you to stay on because I think Sandy and Shipla will move.
Well, thanks, Griff. I really appreciate it.
All right. Take care.